So our last speaker is Beck, and he will talk about order and disorder in biological processes.
Okay. So there is something funny all around here that nobody actually knows, but David and I were the last organizers of the Nano conference. So this is why we were punished to be the last one in the last session. But I know how much work it is to actually organize this conference. So I think that this is a really good time to give a hands for the organizer for doing this great conference. Okay, so today I'm going to talk about order and disorder in biological systems, in biological structures. And first thing that I'll do, I'll follow what was asked and to explain a little bit about what we are doing in my group. So the basic concept about systems that we are studying in my group is studying the relationship of structures on the nanoscale, which is related in somewhat to biology. I'm a physicist, but I'm interested in biological systems just because they present an enormous challenge to the complexity. So the type of systems that we are working with are either protein complexes that forms different supramolecular nanoscopic structures, lipid complexes, lipid protein complexes. So you can see that you can involve a lot of biological molecules in order to make these nanoscopic complexes. And one of the topics that I will talk about today is actually about what is known as disordered proteins. So that one I will go into details. So the basic thing that I started to study biology not so long ago, and there was always for me at least, there was always like a brick wall in between physics and biology. On one hand, if you open up any textbook in biology, the first thing that you learn that there is some order in life. There is a dogma which is very important. And the dogma tells you that since you have a sequence which is encoded into the DNA, this sequence will eventually be decoded into a protein chain. The protein chain contains all sorts of different amino acids. These are different molecules. Due to the interactions between those amino acids, they will fold. I think that Shachar, if he's still here, have shown this diagram, that these structures will fold, that take the protein and will fold it into specific configurations. And in the end of the day, you will have a beautiful three-dimensional functional structure like this famous Nobel-winning molecule of the ribosome. Well, when we think about physics, we think about things in a little bit more simplified way, I would say. And if we take, for example, statistical mechanics, we know that we have interactions in between the entities that we have in the samples. We take that, and we heard the talk basically a few minutes ago, that you need to take some kind of an ensemble average of all the interactions that you have there. And if you plug it in all together, you will get some properties. So these are two different types of way to think about problems. And I think that in my view, I'm trying to find the gap in how to break this wall in between those two disciplines. The first thing that, not the first thing, but one thing that I learned while I started to study biology, that although this dogma is very thought in the first year of biology, it has some problem. It's true that we have amino acid, which is sequenced and or that they have been synthesized, and we have a certain polymer out of that. But 50% of the protein, and this is a lot, right? 50% of the proteins that you have in your body actually have large domains that do not fold, that do not have those alpha helices or beta sheets. They don't have trinary structures for those domains. Some of those proteins completely do not form any three-dimensional stable structure, but they still function. So the basic dogma that we are all building our biology upon is good for many of the cases, but in some cases, it's just not adequately describing the system. And this is just an example of, let's look on this protein. This protein has a well-structured domain that will remain over time for the same configuration. But another domain, which you cannot hardly see, but all of this floppiness domain will just fluctuate over time inside the solution. Right? So this is a disordered or a mixed order and disordered system. And one of the things that I want to do is try to find a way to characterize it, because simple techniques of characterizing mainly depends on the fact that things are ordered. So how do we actually measure order? So one of the techniques that we had a wonderful talk in the morning is using electron microscopy. It's also been used in lower resolution also for biological system, right, in cryogenic conditions. But one of the other things that we learned also in the morning that you can, when you want to learn about structures, you need to take into account interference as a tool for understanding very small structures in samples. And that goes back, if we think about X-ray scattering, goes back to Bragg's law, and we also heard that from Ron. And we can detect out of that interference patterns, and from those interference patterns we can go back and understand the structures that we had before. So if one would have a crystal, he can scatter out of it and get a pattern. And from this pattern, you can actually denote each of the atoms in your protein where its location, up to a very good resolution. The thing is that for doing that, and going from here to the atomic model that you would have, you only are missing one point, and that's to make a crystal. And making a crystal is the big deal for crystallographers in biology today. The problems with those 50% of the proteins that we have in our body is that they do not crystallize. Why don't they crystallize? Because they have floppiness embedded into them. So they will not fit into a specific unit cell that repeats itself. So what can we do? The challenge, in a sense, that we have, in particular for those disordered protein, that the length scale is very small, right? It's nanometric and even sub-nanometric scale. And it's all surrounded by water. And all the interactions, and I'm thinking about the structures as a cause from the interactions in between the monomers, in a sense, or the amino acids, are directed through the media that they are surrounded by the ions, by the water. All of that needs to take into account. But when you try to measure some interactions, when you take out the water, everything changes. And if you put some markers into there, markers themselves are on the same length scales as those interactions, effective length scales in the sample. So what can we do? So one of the techniques is, again, using X-ray scattering, but using X-ray scattering not for crystalline samples, but for samples that are actually in solution, where you have water surrounding you. And if you can do that, you can go for systems that have, let's say, lipid and DNA complexes, or just lipid complexes, or just gold nanoparticles immersed in water. And all of those are measurements that we did in my lab. Or you can go to protein complexes and still have some remnants of the structure. The basic message is that if something repeats itself, either a structure or a location between two structures, we can measure that. We can measure the interference out of it. But the big thing, or the big surprise, is that even if you take something that doesn't have a through structure, and it's all mixed up in solution, you still can have a signal from the interference that was there in the solution, but the interference is very, very weak and very much averaged out into the sample. So the key message is that if you have well-defined order, you have well-defined peaks, and the peaks positions that you see in the interference give you the repeating length scales that you have in the samples. Today we are going to talk about what are you going to do when you have this kind of data. Whether you can still extract some structural information from those kind of systems. So for that we are going to use what is called small angle X-ray scattering. And the reasons that we are using small angle is because we want to probe large length scale, nanoscopic length scales, on the length scales of tens of nanometers. So if you want to go to small length scale, you must go to smaller and smaller angle. By small, I mean fractions of a degree. But the big advantage is when you do such measurement is that it's non-destructive. It doesn't require any tagging. It has very high resolution for very large length span. But it's also suitable for liquids, suspensions, and gels. The idea of the experiment is very easy, and this is the kind of a setup that I have in my lab, is that you take an X-ray beam, and maybe I will run the movie again if you want. You have an X-ray beam that shines your sample, and it can be in water again, and then it just scatters onto a two-dimensional X-ray imaging plate, that from that you can record your data, your scattering data. And you need to make sure that your detector is very far apart so you will have a better angular resolution. Okay. So I told you that SAXS, or small angle X-ray scattering, or scattering in general, is a measurement for the structures that you have. In fact, what it does, the scattering, like any kind of scattering, is a direct Fourier transform for the physicist, or a spectrum power for the biologist, which is the same thing, for the electrons that we have in the electrons or the specimens or the atoms that you have, if you want, inside your solution. So let's just replace this one with a protein, okay? And this protein's, the intensity that I will have on my detector will be just this little type of equation. So if something is very much ordered and repeats itself, I'll have very good signal. But the problem is that when I'm probing protein, for example, dispersed in solutions, they will come in all kinds of configurations. So I need to average out all the possible orientations that my X-ray beam will see while it passes through the samples. This is what is called as powder average, but that's automatically erase a lot of the information that was there. This is why crystals are very important when you don't want to do crystallography. So the question is, what are we going to do if this is not the only problems that we have? I told you that I'm going to work with flexible polymers or flexible proteins, that they do not fold into a specific configuration. Now, it's not that I only have that they all come in different configurations in all different angles in the solution. They also flip between different conformations those proteins are. And the big questions that we have in hand is whether we can get still some useful structural information about those proteins. So generally, using small angle X-ray scattering or small angle neutron scattering was used for half a decade or so in order to study all kinds of systems, usually polymers, because out of those experiments that you will do, you can get some average parameters over the system. For example, you can detect out of that the radius of gyrations. How big is the polymers on average, right? How much space do they occupy inside the solution? You might even get from a small calculation what is the pair distribution, how much one molecule along the polymer see another one out of those scattering curves. But all of those parameters are very global and average of all the different conformations those polymers visit in the solution. And here comes biology to a rescue. Because proteins are not just simple polymers. And this is very important. And the reasons that they are not simple polymers is because the monomers that we have embedded into these polymers are very specific. And moreover, in most of the cases, we know exactly which monomers come after the other one. And they are not all the same. We have the sequence of the proteins itself. So this is well defined. Unlike synthetic polymers, I can tell you that it's going to be exactly 345 amino acids. In polymer science, you will have a very large polydispersity of the length. If it would be 10%, it's fantastic. In biology, it's always the same. So that's really squeezed down the amount of noise or the amount of flexibility that you have in your data sets. The other thing is, not all conformations are possible. Because of those amino acids, they cannot visit in any conformation in space. They are restricted by the fact that they have those side groups and so on, so they can only visit specific conformations. And this is embedded. So you don't need to go over all free space and find what are the conformations, but you are restricted to a smaller space. And the last thing, as I told you, those proteins sometimes have ordered regions into them. Those ordered regions we know a lot about. We know exactly how the proteins are going, how the amino acids are going to be ordered over there. So we can take this information into account. And the question is that, can we squeeze out of this very boring curve of small angle X-ray scattering, can we squeeze some more information about the conformations, but not in the sense that I'm going to have a single conformation, that this is the one that I want to pick out, but about the conformation population. Which conformations are more possible or more plausible in the solutions in comparison to other conformations that are less occupied in the solution? And I wanted to remind you that I'm measuring 10 to the 23 proteins in one shot. So I'm measuring all of those conformations, and what I'm interested in now is to detect what are the statistics, what is the populations of those conformations that I measure. And obviously the answer is yes, because if no, we go to the bus and go home. So, and I'm going to present you one of the methods that we are using. It's called Ensemble optimization methods, but there are some other techniques that are also being used in the literature, and it was all developed in the last 4 or 5 years or so. So the basic concept is that if you take something which is flexible, you can detect out of it some kind of populations of, let's say, the sizes that it occupies, the radius of gyrations in a sense. And it will give you one size of, let's say, one population graph schematically. But if this, let's say this is a protein, and now we'll have a different one, which is a more compact version of that, it will automatically have a different type of populations. If we have, let's say, just those two different intermediates, this is how it's called in biology, two different intermediate sizes which are more populated in the sample, obviously we'll get some kind of a combination between those two populations. And these two populations are very important because it tells us a lot about the system, whether it is flipped from one population to another. So this is what we are aiming to understand. So the techniques is relatively simple. It's unbelievable that it works, but it does. And we try to do whatever we can in order to make it not work, but it just cannot be beat. And the idea is that you define the constraints. You know what is the amino acid sequence. You know where is the structured regimes. Then you plug it in. What are the coordinates of the atoms for these structured regimes. Then you create a random pool. For all of those flexible regions, you let it to be as flexible as you want, and you define out of that 10,000, 20,000 models of different conformations that you might have in space. This is just randomly created. If you have enough conformations, due to the redundancy because of the different types of scattering curves that they will have, that will be enough. And then what you do is that you take your scattering data and the scattering out of that was made out of the pool that you have just drilled out of the hat, and you run it through some kind of a genetic algorithm that try to eliminate those that doesn't contribute to better fits the data and elevates those that contribute more. And you run it through some iteration, you give the computer, and this is why it was done only in the last 5-7 years. Now we have a very good computers. So then you let it run iteratively until it finds a subset of conformations which are representative of the conformations that are possible in the solution. But you don't get the specific conformation doesn't tell you anything, because I will not be able to see a specific conformation. But what I will be able to detect out of that is that I will have distributions of, let's say, the radius of gyration or the maximum extensions of the protein. So this is what you get in the end. You start with the blue radius of gyrations distribution function. This is that we drill out of the hat. And after you run it for a long time, you come out with the different types of populations that was genetically, through this genetic algorithm, was better fitting to your data. And then you get a good, get a good fit, and you can know a little bit more about what are the conditions for what is more possible to be in the solutions of your protein. So let's give one example. This is a collaboration with colleagues from life science, Yoel Hirsch, and with his student Lior Almagor. And the work was mainly done by Ram, my student. And here we studied order and disorder in voltage-dependent calcium channels. Okay. So when I first met Yoel, he came to me and said that, well, we have a really interesting project that maybe we can do together. And this is about voltage-dependent calcium channels. And this is what he described to me. There is a calcium channels. It's a membranal complex that have this protein. This protein is connected to another set of protein and through a hinge. And this hinge, we know that for a heart muscle and for a neurotransmitter release, okay, in the brain, there is only slight modifications in the amino acids. They are the same hinges in between those two things, but the activations of the channels is very much different. And I know how to make those analogs, and I know how to make those proteins, and we want to understand how the slight differences in between them makes the changes in the ion channels. And the other thing that we cannot crystallize it. So it's been thought that it's going to be disordered, so maybe we can do something together. So then I looked at that, and I was, oh gee, I don't understand that. So we drew on the board something that I could, as a physicist, could understand. And so this is my version of the problem. So you have a nunchuck. Okay, you have an alpha helices nunchuck, or this is a stand, and then you have some kind of a flexible region over there, and then another alpha helices or another handle. All right. The difference between the proteins is in the nunchuck, is in the chains in between them. Some of them are more flexible, some of them is less flexible. Why? I don't know. But this is the thing. And not only that, one part of it interacts with a third component. The third component is called GUK. Okay. So, and we want to understand how's the flexibility between those two proteins that have a little bit different chain, how that affects the interactions with this GUK, because it's very important for biologists. All right, so I understand the problem. I want to understand the structural differences in those chains. So the first thing that you do is you say, okay, this chain is very small. There are not a lot of amino acids over there. So it's a very short peptide. It's a very short protein. So maybe it's just one alpha helices. So we try to take the data. We take the scattering data. We just try to fit an alpha helices that will have all, powder average and everything. It doesn't fit. It's not a single alpha helices. So that's thing that we know exactly from day one. But the other thing is that you can do some manipulation to the data, multiply it by, and make what is called a Crafty plot. I won't go into the business. But basically, if you look on this kind of curve, you know that these kind of curves present for you something which is unfolded or disordered, while if you had something which was folded, you will have deep in the Kratky plot, taking exactly out of the scattering data. So there is no manipulation here. This is disordered. Okay. We know that it's disordered. Good. We have something to do. The other thing that you can do is, again, learn about some average numbers that you can dig out of the scattering curves. And this global parameter is the per distribution function that gives you information about it. So when we compare CAV 2.2 to CAV 1.2, and this is when it's interacting with this third player, which is called GOOK, you see differences. Not a lot, but you see some differences in that. But this is, again, this is a global parameters that you have from what is the per distribution for all of the amino acids inside your solution. So you see that there is some differences in between them, but they are very minute. And the reason that they are minute is this GOOK protein is very ordered and it dominates all the scattering. So the next thing that we go is to go to understand, okay, what is the origin of the flexibility? As for that, there is like 100 different types of computer algorithms that you can run and that can give you some predictions for where are going to be disordered regions. Great. We picked up one. I mean, we picked up all of them, but then we, in the end, used one. And we actually find out, so this is CAV 1.2. This is CAV 2.2. This one has a lot of disordered propensity to be disordered. So we said, okay, let's take all of these regions that are not disordered, going to make them alpha helices. Well, let's take all of the yellow parts, which has propensity to be disordered, make them a flexible polymer with all the restrictions for the fact that they are amino acid and not a simple polymer. And let's run it through this algorithm, this EOM algorithm, Ensemble optimization techniques algorithm, and see if we get a better fit than what we had with a simple helices. God and behold, it fits the data. It fits the data quite well. Okay? So we have a very nice fit if we just take these three little pieces to be flexible, and if we take just this one piece to be flexible, it fits the data. Okay? We are happy. But this is not the end of the story. We can get some information out of those fits. And the information that we get out of those fits is what is the populations of the sizes. This is what we wanted to get to understand. And what we get is that both of them has the same radius of gyration. All right? So the average number that we dig out of that was the same. There was no way to detect that. But the population looks much different. The population of one is very sharp and very narrow, and the population for the other one is much more extended. Although that we start with similar initial conditions to run the information. So one of them is more extended than the other one. So why is that? Okay, so 2.2 is more compact than 1.2 in solution, although so this is what we see. We did another thing, and I'll go over that. That's just. So the last thing that I wanted to see was whether we are, okay, I got a lot of regions there to be flexible. Maybe I'm just trying to, try to fit an elephant into a car or something, because I'm giving a lot of flexibility. This is a lot of freedom to pick up a lot of conformation. So, and then we got back to the biologists, and they told us, the fact that they have here, you cannot see that because of the colors, but the fact that these regions are flexible is not really surprising to us. So good, now you're telling me that. And the reason that in each of those locations there is glycine. And glycine is the smallest amino acid, doesn't have any side chains, so this is why it's flexible. Okay, so it's like a hinge. So I said, okay, so this is where you have glycine inside there. Let's take a reductionist approach, and let's try to imagine that we don't have such a big flexible regions. Let's say that we just have hinges, and the hinges will be the glycines. And let's assume that just those are flexible, and let's see if that fits enough, if that's enough to fit the data. So we take the glycine. There are 3 glycine, let's say for CAV 1.2. And so let's try to find the minimal model. Which hinge is the most important one to fit the data? So they are called glycine 436, 449, and 466. All right. So those are the glycines, are the locations. let's assume that just the glycine residues are flexible, all the rest is alpha helices, and let's go for all the combinations of glycines that you have, for them to be just joints, as joints between alpha helices. All right? So you have 3 of 3 of one joint, 3 options for 2 joints, and 1 option for 3 of them. And what we find is that if you just take that glycine 466, the last one, doesn't make any difference. Doesn't make, if you make it flexible or not, doesn't fit the data. So these are just the chi-plots, the fit quality. But if you take the 436 and the 449, it's pretty much already just one of them as a hinge fits the data pretty nice. If you take just 2 of them, that is pretty much the same quality of a fit as I would have if I would take all of these large regions all together. But then that was not enough. So then Ram came to me and told me, well, let's assume that nobody would told us that this is glycine, that it's flexible. So let's look on all 2 hinges in the protein that are possible to make hinges. And let's see what we get. So that had to be run on 7 computers for a week, but I was done. And this is the map that we get. And this is quite beautiful. So this every pixel here gives you the fit quality for, let's say that we take this one and this one amino acids to be flexible. So white is good, and black is bad. And what you see is that, first of all, in these regions that I called the chains in the beginning, this is exactly where we have the best propensity to fit the data. But the last thing that we see, that the best of the best were along a line. So along a line gives you the differences between 2 amino acids, right? And let me tell you that the difference along the line were exactly 13 residues apart. And if you don't want to take out your computers, that 449 and 436 are exactly 13 amino acids apart. But that was a really nice show that even if we picked an amino acid, we don't have amino acid resolution, but we can actually find a good indications that those are actually in amino acid the resolution that are most likely to be flexible. So I will just say, okay, yeah, that's nice again, but we don't have time for that because David is standing, and I like him a lot. So I'll just give you the take-home message that flexible protein can be measured in solution. And we know that CAV1-2 is more flexible. And we also, this is something that I didn't show you, that we can also measure and see when it's interacting with a third partner, how much those conformations are actually being changed while it's interacting, because we can do that through the same kind of a technique. And with that, I would just want to thank my great collaborators, funding agency, my great team that I have, and just click this one to thank you.
Questions?
Who wants to go to the bus?
Either in your calculations, so are the flexible sections weighted by energetic calculations of relative stability?
No.
No use of doing that?
People do it, and in the end of the day, I mean, you can run like Monte Carlo or molecular dynamics to do that. It just occupies your computer for a much longer time. You cannot even run it on your computer. You need to run it on servers. And in the end of the day, you end up with the same thing. So the basic concept is that if you draw out of a hat enough conformations and you wait and then you run it through this algorithm to see which one will be more possible to be selected, they will eventually find their way to discriminate those that are not energetically favorable from the data set. People are doing that to run molecular dynamics for such a large data set. It just takes a lot of time, and it doesn't give you much more information in the end.
Can you say something about the time resolution of the method? Would it be possible to probe some kind of dynamic process?
Well, people are doing that. You need to remember that for that, you need to do some kind of a synchronization, right? Because what I'm measuring, what I showed you now is just measurements of all different conformation that was measured in the sample. So there were, like, again, 10 to the 23 conformations that were measured once. So, but there are techniques using, let's say, microfluidics that you initiate some kind of conditions to go from a certain structure to a second structure, and the dynamics can be resolved if you just push it through channels and then you pick every point in the channel. It gives you, it's been translated into time series, right? Because when two things interacted or two things started to mix, and after a while they just go through the channel. So that can be measured with a microsecond. It's pushing it, but millisecond is very easy. But also in the microsecond regime, it can be measured for going from one structure to another.
But if there's an aggregation process, for example?
Oh, this is the thing that we fear the most. So, and aggregation we see beautifully. It's just destroy all the data. So it's so that's the thing that terrifies us.
Can you help the algorithm by changing the sequence, like replacing amino acids?
I can do in the algorithm. Yeah, I can do it. But what it makes any sense?
No, in the algorithm, simply produce another protein with different sequences.
Yeah. I mean, this. Well, yeah. Of course. I mean, you're talking about whether I can change my proteins, doing like some kind of a mutations, and then see
like
how
you assume that this is the point.
That's the thing. This is what well, in a sense, we didn't do the mutation. We let nature do the mutations, right? Because we had two proteins that were very, very similar one to another, but one of them had one glycine, and the other one had three glycines. Okay, and we wanted to understand why. Okay, why did nature make them so tinily different, but still they have very different activity. Okay, so then this is why we didn't do it for one. We did it for those two. And obviously one can do, and this is again something that is known, that when you do mutations to those glycines, you have terrible diseases. Okay.
But you can do it in a consistent manner.
Yeah. I mean, expressing proteins with specific mutations is day-to-day work for biologists. So this is something easy to do.
And how well is it conserved?
Yeah.
So
Yeah, 100%. That's the beautiful thing about it. That's the beauty of biology. I mean, you don't need to worry about these locations. I mean, they are pretty much accurate. Yeah.
But it's a conserved region across families of
Oh, yeah. This is a very conserved region. Yeah.
So I have a question about your experimental setup. So I have a question about the experimental setup. Can you get more information if you use a polarized X-ray? And also if you can measure the polarization of the scattered...
Well, no. Well, yes and no. Not just polarized X-ray. Wow, gee. How can you do that, and why would it help you? You can have more coherence, and this is what people are doing now, right, with a free electron laser, to have more coherence into the data, and by that getting way more information because you're more sensitive to the phase. But not for small angles. It's mainly for type of reconstructions through reconstructions of the phases. Polarized one. I don't know how you polarize the X-rays. That's to be honest. I don't know the lens. I mean, you cannot just buy those lenses, right? I mean, this is pretty. Well, yeah. I don't know. We need to think about it. Yeah. What? Yeah, it's kind of polar. I mean, yeah, it's true. So in synchrotrons, you have polarizations embedded into that. I mean, we need to take that. I mean, this makes a whole lot of problems for us, but never mind. So in synchrotrons, where those measurements were actually done, some of them was done in my lab where it's not polarized, and some of them was done in synchrotron where it is polarized. Doesn't really make any difference because all of the samples are just jiggling around. So it sometimes you see this polarization, sometimes you see the other polarization. It doesn't make a lot of difference for those kind of materials. It does for metallics and so on.
Okay, if there are no further questions, let's thank Roy and the rest of the speakers in the session.